Hello everyone. Today we'll discuss the topic 4.2 blood clotting intrinsic and extrinsic factors. When blood is shed out or collected in a container, it loses its fluidity and it becomes a jelly-like mass after a few minutes. And this process is called as a coagulation or clotting of blood. The clot is a mesh of thin fibrils entangling the blood cells. These fibrils consist of fibrin and the fibrin is formed from the fibrinogen. The This blood is normally the fluid while circulating in the blood vessel, but when the blood is punctured or blood vessel is punctured, the blood flows out of it, and the blood after coming out or coming in contact with external environment, it loses its fluidity or its liquid nature and it becomes a jelly-like substance. The process is called as a blood coagulation. The blood clotting or coagulation occurs in three main steps. Generation of plasma thromboplastin, formation of thrombin from prothrombin, and formation of fibrin clot from the fibrinogen. There are several factors which are involved in the blood clotting, which occurs through a series of reactions due to activation of variety of substances. These are factor 1 is a fibrinogen, factor 2 prothrombin, factor 3 is thromboplastin or tissue thromboplastin. Then factor 4 is a calcium ion. Factor 5 is proxylerin or labile factor. Factor 6 is missing, which was later found out to be same as factor 5. Factor 7 is a stable factor. Factor 8 is an anti-hemophilic factor. Factor 9 is a Christmas factor. Factor 10 is a Stewart-Prover factor. Factor 11 is a plasma thromboplastin antecedent. Factor 12 is a Hegman factor and factor 13 is a fibrin stabilizing factor. All these factors are written in the Roman numerals from 1 to 13. These are some coagulopathies which are related with the deficiency or the defect of the these deficiency or the defect of these factors in the blood. Say for example, the defect in the factor 8, that is the called as a hemophilia A. Factor the hemorrhagic defect in the factor 9, that is a hemophilia B. Then 11th factor hemorrhagic defect in the 11th factor, that is a hemophilia C. And the last, that is a what von Willebrand factor, that is von Willebrand disease, that may be due to this. The intrinsic and extrinsic factor, the all these both the pathways, the three basic steps are there. That is, the first is the formation of the prothrombin activator. Then second is the conversion of prothrombin into the thrombin. And the third one is formation of the fibrin clot from the fibrinogen. The sequence of clotting mechanism, the normally during circulation the blood does not clot but because the enzyme involved in the clotting are in inactive form. Slight initial activation causes clotting in which each enzyme activates another one in a sequential manner till the conversion of fibrinogen into fibrin. In general, the clotting occurs in three steps as we have earlier discussed. The formation of this prothrombin activator, then second one is conversion of prothrombin into thrombin, and the third step is conversion of fibrinogen into the fibrin clot. During the process of blood coagulation, the clotting factors which are in inactive forms are converted into active forms, and their enzymatic action produce the successive reaction one after another in a cascading manner. Thus, the various reactions involved in blood coagulation are explained by enzyme cascade theory. The two types of the or the these blood coagulation occurs in a two ways. One is an extrinsic pathway and another one is an intrinsic pathway. In both the pathways, the second and the third step remain same, but the first step, that is a formation of the prothrombin activator, is different in extrinsic as well as intrinsic pathway. In extrinsic pathway, the formation of prothrombin activator is initiated by tissue thromboplastin. In the intrinsic pathway, the formation of prothrombin activator is initiated by platelets which are within the blood itself. This is the intrinsic system, and you can observe this starts from the what inactive factor 12, and this inactive factor 12 it gets activated into the what which form that we called it as a what 12a. That this is the 12th factor is a what inactive factor, and this inactive factor it becomes that 12a, that intrinsic and it is in presence of the high molecular weight, that is kininogen and the calligrade. This activated 12A, which is formed that converts the inactive form, that is a inactive 9 in factor into the active, sorry, not 9, that is a inactive 11 factor into the active 11A. And this active 11A now it acts on the inactive 9, and this inactive 9 gets now activated. This now activated 9A acts on factor 8, that is an inactive one, and this 8 factor become activated, that becomes 8A. Now this 8A it acts and in presence of the what phospholipids and the calcium ions. This 8A is now acting on the factor 10, which is an inactive one, and it converts 10 factor into the active 10A. Now this active 10A, then phospholipids, then the calcium ions and 5A. These four things, that is factor activated A 10A, factor activated 5A, phospholipids and the calcium ion, that is a factor 4. All these combine together to form a prothrombin activator, and this step is these all are the steps, that is the formation of prothrombin activator by the intrinsic mechanism. Means in the intrinsic mechanism that starts from the factor 12, inactive factor 12, that gets activated and it becomes a 12E. This 12A then activates the 11 into the 11A, then 11A activates the inactive 9 into the active 9, then in active 9 it acts on the 8 inactive to form the 8A active, and that 8A active activates the factor 10, which is the inactive one, into the active 10A, then active 10A in and the phospholipids, calcium ion and the factor 5A, which is the active one, all these combine together to form the prothrombin activator. In the extrinsic system, the this is a tissue thromboplastin, that is it starts from the factor 3, that is the tissue thromboplastin. It this tissue thromboplastin which generated due to the what the tissue damage, it activates the inactive 7 into the 7A. Then in presence of the calcium ion, phospholipid and the tissue thromboplastin, it activates the factor 10 into the 10A. And this 10A, after that, this active in presence of the phospholipid, calcium ion, and the 5A, it forms a prothrombin activator. Means in the extrinsic system, if you observe, the only the what the tissue thromboplastin, that is a factor 3 and the factor 7, these are involved in the generation of the prothrombin activator along with what that is a factor 10 and the 5 calcium ion phospholipid, all these. But in intrinsic system, the factor 12, factor 9, factor 8, factor 11 and factor 10 and the factor 5, all these are involved in a cascade mechanism. The tissue trauma or injury to blood vessel or damaged endothelium exposes subendothelial collagen, that is the von Willebrand factor. Platelet adhesion is the process by which platelet attach to the exposed subendothelial van Willebrand factor. These platelet, it undergoes the two events. First one is a platelet will undergo an irreversible change in shape from smooth disc to the multi-pseudopodal plugs, which greatly increases their surface area. And second, platelets secrete their cytoplasmic granules. As we have earlier discussed, in an intrinsic pathway, injury to blood vessel or the tissue trauma, it causes a activation of the factor 12. This factor 12 gets activated and is in presence of the what kininogen and the kininogen or the high molecular weight kininogen. The factor 12 which was activated factor 12 in presence of the kininogen and the prekallikrein, it activates the factor 11 and the factor 11 it becomes activated one. This activated factor 11 in presence of the calcium ion, it acts on the factor 9 and it forms a activated factor 9. And this activated factor 9 in presence of the calcium ion, it causes or forms the activated factor 8. And this activated factor 8 where that acts on the factor 10 and it activates the factor 10. Now this factor 10, then this factor 5, calcium ion and the phospholipids combine together to form the prothrombin activator. What are the prothrombin activator precursors? That are the activated factor 10, activated factor 5, calcium ion and the phospholipids. These combine together to form the prothrombin activator in the intrinsic pathway. In intrinsic pathway, this factor 12 is converted into the factor 12A, and in presence of high molecular weight kininogen, this factor 12A converts factor 11 and inactive 11 into the active 11A. Then this active 11A, it activates the inactive 9 into the active 9A. This active 9A now act on the inactive factor 8 and it converts it into the active factor 8A. And this active factor 8A in presence of the phospholipid and the calcium ion, it acts on the factor 10 to convert it to a 10A.